Coming up, another round two of my conversation with chemist and computer scientist James Tour. Our conversation yesterday was so interesting. I wanted to go in depth on these issues of origin of life, the compatibility of God and science. So we're going to get into it once again with Professor Tour. Hey, if you're watching on YouTube or Rumble or listening on Apple, Google, or Spotify, please subscribe to my channel. This is the Dinesh D'Souza podcast. My conversation yesterday was so fascinating with Professor Tour that I want to continue it and now focus on something that you, Professor Tour, have thought about and talked about, and that is the debate over the origin of life. Now, I'll just say that, you know, just from my college years and limited exposure to this debate, I have read that in the 1950s, there was a series of experiments done by, I think, Miller and another guy named Urey. Yeah. And they were mixing a bunch of chemicals together, and they came up with some chemical cocktails and said, oh, gee, well, life must have begun somewhere on the earth in a warm pond. That somehow these chemicals must have come together to produce at least life itself or the beginnings of life. And then separately, I read, I think this may have been from Francis Crick, the biologist. He said, no, that's all nonsense. Life must have been seeded on Earth from another planet. And again, applying the logic that you did yesterday, you've got two radically different observations about the origin of life. Use those as the springboard to lay out what is the shape of this debate now? Because what I'm talking about is actually theories now from several decades ago. Where does the topic lie now?
Okay. Well, let me give you the bottom line, and then we'll work our way back. The bottom line is scientists are clueless on the origin of life. Now, they will argue with me when I say that, but they are really clueless. You start asking them, and everything falls apart. And the experiments that they do have been so guided that it's the way that would never happen in an early Earth. But as far as the little pond, Darwin mentioned the little pond, but that dates back to the Babylonians. And out of that same pond that people came out of, their gods came out of. So, this is the origin of the little stinky pond.
You're saying it's not, in a way, not even a scientific concept. It has its roots in Babylonian mythology, I guess.
Correct.
Yeah. Okay.
That's the root of it.
Wow.
So when people have said that this is some good model, this is the root of it. But Miller-Urey was not a bad experiment. Actually, it was really quite amazing. Where you put this electrical discharge simulating lightning, and you have to somehow pull these intermediates out of the medium quickly, or they're going to decompose. And so you set it up where things could distill around, and very sophisticated experiment. And he ended up with, there's 20 amino acids, canonical amino acids that we're made out of. There are a few others that our bodies incorporate, but 20 is the canonical ones. He ended up with something, he characterized 5 or 6, but people have gone back and seen that there's really a dozen or 15 of the canonical amino acids of the 20 are in there. That's a great experiment. This was in the 1950s when people really didn't know how to do this kind of stuff. But the other side of it is you make hundreds of other amino acids which our bodies never use. And they're not just alpha amino acids, which is where is the nitrogen, where is the amine relative to the carboxylic acid. All of these canonical ones are alpha amino acids. But you get beta amino acids, you get gamma amino acids, and nobody ever talks about these. You could never use anything from Miller-Urey's experiment and start trying to incorporate that in a living system. It's way, way too impure. So nobody ever uses it. It's so impure because you get hundreds and hundreds of amino acids. None of them have anything to do with anything, and they would be totally unusable and mess everything up. They never talk about this. So mixtures is what these people make. I mean, their middle name should be mixture. And so we have tools in chemistry that can detect to 0.00001% yield. So that, we made it, but it would be unusable. So for example, you can take supplements in the morning, a whole fistful of them. You take it, your body can segregate these, and it knows what to do with each one of these because you have enzymes. And you can't do chemistry like that. You have to use really pure starting material. So you take pure A, you mix it with pure B, and you get C. If you have impure A, where A is 90% pure and B is 80% pure, so now you have 30% of other stuff in there. Those are going to compete because very often the impurities in A look a little bit like A, and B will start reacting with those. The impurities in B look a little bit like B. And so this is always what these people generate in their experiments that they're so-called simulating early Earth environment. So everything they have ever made is useless. They almost never take what they have made and bring it on to the next step. They will identify that it's there, then go to a chemical company and buy the pure chemical that's been made by others some other way or isolated from a natural source. This is called relay synthesis. And they do it at step after step. I could never publish a synthesis in a paper on that. You have to go from A all the way to Z. If you do a relay, you have to say, I did a relay, and people discount a lot for that because it's not a real synthesis. So from the base, everything is cheating. That's on top of the fact that everything in the Miller-Urey experiment was racemic, which means that every molecule in every one of the amino acids, canonical amino acids, except one of them, because it doesn't have a stereocenter, comes in a handedness. You get a left hand and a right hand. And nature only uses one. With all molecules, it only uses one and not the other. And this had both. And you can't do the chemistry when it has both because it messes up everything. The proteins wouldn't fold right. Everything is wrong. And this whole thing, well, maybe it did reuse mixtures and later on evolution took care of that. We know by more recent work, just in the last 25 years, because of something called chiral-induced spin selectivity, you had to have high degrees of homochirality from the beginning for these reaction chemistry to work. So I'm telling you, Dinesh, at every experiment that these people have done, you can put out 100 reasons why that could not have worked on the early Earth. All you learn from it is, oh, okay, that's how it could not have happened. If you do not believe in the physical resurrection of Jesus Christ, send me an email, tour@drjamestour.org, and we will get together, and I will share with you about why I embrace the resurrection of Jesus.
Let me reformulate what you're saying only to make sure I grasp it, because
Okay.
some of it is clearly over my head. I think what you're saying when you used earlier the term guided is you were saying in nature, you have molecules and atoms and non-living objects, let's just say lying on the ground or floating in a pond, and these are not guided. They are inert, immaterial. They are material objects, but they are not purposefully aiming at anything. They're not trying to become life. They don't have any intentionality to them. That's a stone. That's a pebble. That's a branch, or not a branch, but these are just objects out there. What you're saying is that the scientists that are trying to create the conditions of early Earth and show that life evolved are deliberately acting in a way to produce a known result. And so they're like, well, we need a little bit of this. We need a little bit of this. Can we make an amino acid? Can we make an enzyme? Can we do this? Can we do that? And you're saying these are too radically different projects because nature clearly wasn't functioning with that kind of intentionality. Was that a correct inference from what you were saying?
That's a partial understanding.
Okay.
Yes, molecules have never been known to move toward life. They have no brain. They don't know what they're going to. They have no idea. And unless there is some entity acting upon them, they'll never move toward life. And normally that entity is a human being which tries to push them toward life. And even then, they've never been successful. Nobody's ever made life. But what I'm also saying is every experiment that human beings do to simulate what happens on an early Earth gives them crap. That's what I'm saying.
Yeah.
It gives them something that is totally unusable, and they'll identify one little bit of something there and say, success, that's the molecule we wanted to make. But it would be unusable in chemistry. In biology, it would be usable because your enzyme can find that. Your enzyme can locate that. Now, generally, you don't want to present it with too much garbage because it's going to end up killing you. But enzymes know what to do with things because they're like the glove, and they're looking for the hand that fits in it. But before you have biology, before you have the enzymes, you can't do chemistry on gross mixtures. And here, how many steps would it take to go from water, carbon monoxide, carbon dioxide, maybe a little bit of methane, maybe a little bit of ammonia, to the complex organism of a cell? How many steps? It's got to be thousands of steps. And what happens is the impurities multiply. We had a project from the National Science Foundation 25 years ago, more than that, 30 years ago, that was, we tried to do what DNA does. From DNA, what DNA polymerase reaction does, you take a piece of DNA and you copy it. And then you separate them. So you have the mother and the daughter, and they're the same. And now you copy those, and then they're the same. And this is why they find one little bit of blood, or they find a tissue where somebody blew their nose, and they can go and express that now, and they can duplicate that DNA and say, okay, we know who committed this crime. Right. Because you've done DNA polymerase, which Kary Mullis came up with that chemistry. He won the Nobel Prize for that. And so we try to do that with non-biological molecules. And what killed us was fidelity. In other words, we had 70% yield. We had 30% junk. And then that 30% starts duplicating itself. So the junk starts duplicating itself. And so what happens is, before you know it, it's all junk. Are you saying that the DNA multiplies 1-to-1 without junk, whereas in the other cases, you get a partial copy? You get partial copies. And those partial copies make copies of themselves. And so after a few rounds, everything is unusable. Nature, biology knows how to deal with this. If you look at the yields in biology, it's just extraordinary. They're getting 99.999% yield. And the impurities that are left, you have enzymes that go and find those and get them out of there. Or else it's going to incorporate in our DNA and we're going to get cancer really quick. And then when one does incorporate, you have other enzymes that run down the DNA strand, and they even from 100 base pairs away, they can identify that there's something wrong down there. Boom, they move down, they find the mistake, they pull it out, and they put the proper one in. It's extraordinary. But chemistry is not like this. Chemistry doesn't have these mechanisms. And so everything that they have ever made is junk and unusable. Ask them, oh, you made that? Did you ever use that for the next step? Oh, well, to streamline things, we bought this. So that's what I'm saying. Not only is it the human input that you're putting in at every phase. What if you didn't go in your laboratory today? What if you didn't mix those? Would they mix by themselves? There's an earthquake in California. Did they mix by themselves and just get shaken together? I mean, this is human input, which wasn't around on an early Earth. But then even with all the human input, they make junk. They make junk that's totally unusable, and they claim that they got it. So much so, I'm telling you, Dinesh, these guys have claimed we're almost there. These guys have claimed, Jack Szostak, Nobel Prize winner, Harvard, said in about 2014, we will have life in our lab in 3 to 5 years. This is crazy. And then a few years later, he confessed, well, we can't even quite make the RNA. From an RNA to life is a universal way. And he can't even but he made that claim. And then Dimitar Seslov, who was also at Harvard, said the same thing at the same conference.
You described, I think, beautifully in one of your talks, the strategy these guys use, right? In other words, they'll focus on making one element. They go, we got this. And then they rely on the media to take the claim, which already is exaggerated in technical terms, but then to blow it up into some headline that is divorced from what the experiment really was. And then you said the cartoonists step in and then give cartoonist renditions of the story. And this is called science. And I think there are sincere people who look at this stuff and go, these are the people who say things like, well, I'm following the science.
Yeah, and it goes into textbooks.
Right.
Like your primordial soup, your little pond model, that's taught in elementary school. The same model is in advanced textbooks in the university. Not just introductory, advanced. The same primordial. Where did you get that? Lee Cronin said to me, this is a good model. I said, what makes it good? How? How do you go from small molecules to assemble to get a cell? And then that cell go to a higher order organism that comes crawling out of that. How? Give me the steps. On what basis is that a good model? Crickets. Nothing. And this is still in our textbooks to this day in the advanced level. And it just propagates. And so these ideas get into the textbooks and they're all nonsense.
I think part of the reason people get suckered into these kinds of claims is because they do not have any sense of what a cell is. I'd like you to talk a little bit about, because the cell is the root of life. Am I correct? And
yes.
So the cell, whether we're talking about a plant, a porpoise, a chimpanzee, or Dinesh, we're made up of cells.
Every life.
Every life is made up of cells, and the same type of cell, correct? And when I say the same type of cell, I mean cells are the root of all living.
Yes, there's a lipid bilayer on the outside. There's some DNA, and yeah.
So what do we know? What can you tell us about the cell that gives us a sense of how it functions?
Yeah, the cell is a factory. Because of you, millions of people have heard the gospel. If you confess with your mouth Jesus is Lord and believe in your heart that he's risen from the dead, and you will be saved. That's right. That's the requirement. We talk about science concepts which draw people in. Take these nanomachines and have them drill into cells. This would be a great way to kill cancer, right? We also talk about Jesus Christ, who's the best in everything.
My faith in Jesus Christ means more to me than anything.
If you could continue to give or give for the first time, we would certainly appreciate it. You can go to jesusandscience.org. Thank you so much. So 130, 140 years ago, it was just protoplasm. Even 100 years ago, it was just protoplasm, and simple little thing, and just a bunch of protoplasm. No, the cell is a factory. And first of all, it has information. It has the computer chip that's running everything, the informational code. That is the code. And that's coded in DNA. The sequence there is the code. In other words, just having a box of letters, there's nothing. There's no book there. You have to take those letters, put them into words. Now you have a code. Now you have a book. But just to take the letters randomly, there's nothing. So there was inherently a code that's inscribed in the DNA. The DNA has all this machinery of enzymes around it that will duplicate that into RNA. That RNA then will go ahead and build the proteins, which are the little nanomachines that build us, these enzymes. They're proteins, They're enzymatic. And these are the little machines that build us, so that when you eat something, they come and they take those parts and they start assembling your body with that. This is amazing. And all of this is happening inside cells. All of this chemistry is occurring inside the cell like this. And so now to try to build this, and how do you get, if you go to a factory, how do materials get from this side of the factory to this side? A lot of times you see these overhead transporters, someone picks it up and it's carried, and this is exactly what a cell has. To move material from one side to the other, there are these tubule-like structures that will form. It needs something from over here, so it builds a tubule, and things will come across this to go to the location that's needed. And then the tubule will decompose, and they'll build a new. Well, why didn't you leave the tubule? Because you could use it again later. Because then the cell would become too rigid. It'd have tubules all over. So it just builds them, and then it takes these components and breaks it apart and rebuilds another place. It's fascinating. There's so much in the action of a cell. You have all of these subcomponents. Now, people will argue. They will say, well, you're talking about a modern cell. The simplest cells at the beginning were different. Well, this has already been calculated. First of all, the fossil record, if you look in the fossil record, the simplest cells that we can find in the fossil record are about the same simplicity, the same level of complexity, I should say, as our simplest cells today. So we don't find anything simpler than the simplest ones we have today, because you need a minimal set of items to get this thing to run. So biophysicists have calculated what is the simplest cell you could have that could operate. And these cells would be so simple that all of the amino acids that they would need, they'd have no ability to make them. Everything would have to come from the outside. For us, we have the ability to make a lot of our own amino acids. Other ones are essential that we need to eat. We don't eat, we die. And this cell couldn't make anything. Needed everything delivering from the outside. How you get all these amino acids from the outside to it, that's that cell's problem. But just talked about the simplest cell. It had about 15 different components that would be needed. DNA transcriptions, RNA, and so you'd need all of these different components of things. Of those components, how many of those have been made in origin of life experiments? You know how many? 0. None. Of those 15 components, do you know how many of those have been made, not just under a rock, in a laboratory with modern methods? When you're talking about these subcomponents, I'm not talking about DNA. I'm talking about these higher level structures that you know how many have been made in the modern lab? 0. Unless they've taken it from a living cell. You decompose a living cell, you take it. So nobody's ever made any of these. How on earth can they be claiming that we're very close? Steve Benner, another origin of life investigator, has said most of the paradoxes to make life have been solved. And I've challenged him that he doesn't say this anymore, because I've challenged this guy. You're not seeing Dimitar Seslov saying this. You're not seeing Jack Sostek saying it. You're not seeing Steve Benner say this, because I've challenged them on this, and I put it repeatedly on the internet, them saying this. Over and over again. So they backed off, and and Lee Cronin as well, who said in 2011 he'd have life in his lab in 2 years. Everybody misses the deadline, and because it's a hard problem. So you got to be careful about saying this, and the press, whenever they've said it, has picked up on it and run with it. I've spoken a lot. You go ahead and ask me some more questions.
Well, I'm thinking of something that the biologist Richard Lewontin of Harvard said many years ago. He was talking about the idea of materialism. And by materialism, he means obviously not being materialistic and wanting to go shopping, but rather the idea that the universe is made up only of material stuff and nothing else. And he essentially said that our commitment to this materialism is dogmatic, meaning this is our starting point. He goes, the reason it has to be our starting point, he says, is because if we don't take it as our starting point, and I think I'm quoting him pretty closely here, he goes, then we're letting the divine foot in the door. So to prevent the divine foot in the door, even if our explanations make no sense, we've got to deliver them in purely materialistic terms, because that is our axiom. That's kind of like, the axioms that Euclid began with. There's only one line that can go between 2 points. We're going to start there. We're not going to question the axiom. So I think this is what's going on. If they come up with nothing, they have nevertheless got to posit that there is a way, even if they haven't found it. And then to maintain confidence in the scientific enterprise, it always has to be right around the corner. And I wonder if you think some of that's going on here, that because you've got intelligent people here, they know what they don't know, and yet from your own quotations, they're saying things that aren't true. And so I'm just trying to probe what is behind them making these dogmatic assertions unsupported by actual evidence.
As far as what is behind them, you would probably know better than what I know on this. But it is, yes, when what's happening, and I see it, is they're coming around because they're stuck. So there was Lex Fridman's podcast. He had Lee Cronin on there, and Lee Cronin, he said, so people make one chemical, another chemical, another chemical. And he was saying these origin of life experiments aren't getting anywhere. And he even used the word, it's a scam. He used the word. Now, I don't want to quote him on that because he's asked me not to quote him on that anymore. But I was just trying to say, that's how bad it was. Then so Lex pushed him on that. Tell me, he says, because they make one compound after another. But then what? If you take a cell, the cell got its information from somewhere. You've got to have the informational code. It got its informational code from its mother cell, from the thing that it broke. The cell divides, and then it was divided. He says, you trace that back to Luca, which was the last universal common ancestor, that progenitor cell from which all other cells were made. He says that had a code. But that code had to have come from outside the cell. Lee Cronin said that. Why did he say that? Because he's seeing what we're seeing. When you follow the evidence, you're going to see there's a code. There's a code that prescribed for this, and that code must have come from outside the cell. And so now he's got this new theory called the assembly theory, AT, assembly theory, and he talks about causal history. You can take a molecule and you say, could that molecule have formed randomly in an explosion on a meteor? If you find one, maybe it formed random. But if you find 5 of them with that level of complexity, my assembly theory can tell you that couldn't be random. That had to have had a causal history. Now, he needs a big theory for this. I can look at a molecule and tell you, if you have 5 of those, this didn't happen by chance. I mean, something's going on here. But why does he use the word causal history? Because he'll never go near the word designer. He'll never go near. But causal history, I mean, what do you mean causal history? That means that there was something here that did that. And so what's happening is, Dinesh, it's a very interesting thing to watch these scientists. I've pushed them for years. And, at first they'd attack me for God of the gaps. And I never bring God into this discussion. And then they'd attack me personally. And but I keep pushing them. But they will never contest with me academically. Never. None of these guys will. Because they know I'm right. I've even said to Steve Benner, I'm right. I'm right. You see exactly what I see. But they've invested 40 years of their careers saying this stuff. So it's hard to backtrack because scientists have to allay our fears just like everybody else. But what's happening here is they're coming back to the same point that we're saying. The same point that this Harvard professor was saying was dismissive of. You can't just dismiss it because you're going to end up coming back to it.
It seems to me that modern science is both in biology and chemistry on the one hand, but also in physics, arriving at the same kind of point. And here's what I mean. If you look at physics, the idea here is that the universe had a beginning. Now, true, there are people who will posit multiple universes and so on.
That's going away, actually.
Yeah, but that's right. Exactly. Universes with their own laws and so on. But frankly, even if there were multiple universes, you will not have answered the question of where do they come from? Mm-hmm. But what modern physics is grasping is that there does appear to have been some built-in order in the universe to get a universe that has laws, to get a universe that follows laws that Einstein and Newton and others then spend their lifetime trying to excavate, because these laws are somewhat hidden, right? They're not even obvious. E equals mc squared is not obvious. And I think what you're saying is that similarly, when it comes to the origin of life, we can work our way back. We've got all these cells. We work our way back to the original cell. But then we are faced with, and you use the analogy of a factory, but I think even a factory doesn't quite do justice to it, because a factory one can think of in purely mechanical terms, right? You just have pulleys and things moving around. But you're saying in addition to the factory element of it, there is a kind of code, as you put it, or intelligence element to it. There's information that is also organized. Am I right? And so you're saying, and so you have the original cell, and it already has all that. That cell did not evolve. And so modern science is not any closer now than it was, let's say, 50 or 75 years ago to accounting for that.
Yes, that's what I'm saying. And that information is in every polymerized structure in that cell. It's not just DNA. DNA has a code. DNA is a string of letters put together to give you words, to give you a prescription on how to build a cell. That's what it has. And then segments of that book, will be duplicated into RNA. So RNA now knows what to do. So now RNA builds the enzymes that build us, the proteins. Proteins are, these enzymes are all, the vast majority of enzymes are proteins, not all of them, but it will prescribe this. So now the code is inscribed in the enzyme. The enzyme hooks together sugars. The sugars bear that informational code now. So it turns out, because sugars have multiple tentacles that DNA doesn't have, you can put far more information stored in carbohydrates and sugars than you can in DNA. And this is what happens. This is how a cell gets information from the outside. It's not just stuck with the information that it has. The proteins and the sugars display outside on the cell surface. Other cells come up and they will share information through these structures and modify the structures. Now you've got modified structures coming into these cells. And this is this whole idea of epigenetics. Things that come into my body from the outside start affecting my DNA. And anytime we've had a virus, viruses will often put their DNA into our DNA. So we have a history of the viruses that we've had, because those viruses have inserted some of their information into our DNA. But it's not just DNA modification. You modify the enzymes on the surface, and this is going to affect regulatory things as well. So information comes from the outside. Bacteria are even more insidious. They actually, in these biofilms, they will share DNA between them. So in other words, you get a particularly resistant bacterium, and the drugs don't kill this bacterium, and it starts sharing its DNA with these other bacteria, and all of a sudden, they now become resistant. And remember, bacteria populations double every 20 minutes. So you go 1, 2, 4, 8, 16, 30. Every 20 minutes, it doubles population. That's why you feel good now. And an hour later, you're like, I'm so sick. Yeah, because that thing is just doubling every 20 minutes. So anyway, this is the informational code. This is fascinating. And we have no idea where the code came from. And you're right, the machinery in a cell, molecules don't do this. I have built some of the most sophisticated nano machines, molecular machines in the world. My group. We've made nano cars. We've made nano trucks. We drive the nano cars. These are single molecule cars. They're
I got to say, I had a major chuckle. You were talking in one of your speeches about the nano car race.
Yes, we had a nano car race. Yeah,
and
how
you came in first.
Yes.
What was your speed?
We were going. I don't know. It was like a few nanomiles per hour or something like that. Yeah.
Right. But you said that may sound slow, but you were like, that's like lightning fast compared to the guy who came in second.
Yes, we completed the race in an hour and a half. The Swiss came in 5 hours behind us, and nobody else could complete the race in 30 hours. So it was very fast, and we can go, maybe, I don't know, 10 car lengths per second, something like that. That's a pretty good clip. So and these have fully rotating axles. We've built loading bays, so we know how to build. This is child's play compared to what's done in a cell. A cell's machinery, a cell's molecules don't just do this. Biology is amazing, utterly amazing, and the biologists don't see it because they grew up there. It's like a rich kid growing up in a rich home. He thinks everybody, drives a Mercedes. I mean, it just doesn't know. You take it for granted. To somebody like me who has to build molecules from the bottom up, you look at what biology's done, you're like, this is amazing. How did this happen? Every synthetic chemist sees what I see. The biologists can't understand what I see. Why we are so clueless on the origin of life. But Dinesh, they're coming around. They're seeing that there's something here. Lee Cronin, to his credit, is looking at this stuff. I don't see him pushing his stupid little bubble theory that life's going to come out of these bubbles in his lab. He's seeing that Luca, this first cell that is the progenitor of all life, must have had the code from the outside. This is obvious, but these guys wanted to somehow say the code somehow developed on its own randomly.
I mean, in a way, when I mentioned at the beginning, Francis Crick, and the idea that maybe aliens of another dimension brought life, I mean, preposterous or far-fetched though that is, in some ways it's almost like he knew what you know.
Yes.
And he realized that the other type of explanation, that you could somehow cook this up, let's just say in a warm pond, well, it's not going to work. You're going to need some other explanation, some other cause.
Right. But his explanation just begs the question. It's always, where did that life come from? What we're actually talking about is not origin of life. We're talking, what's the origin of first life?
Right.
Then it pushes it back. Where'd the aliens come from? What's the origin of life? Now, as a believer in God, and this, God is the source of the code here, and God is the one who has done this. But I never bring God into this. I'm just saying, look at the science. What does the science tell you? And I've always said that I think one day we will learn how to make life. I don't know. Nobody will ever know how life started on this earth. No one, because nobody was ever there. But we can make conjectures based on experiments how it might have happened. But can we even make it in a lab? Let me distill this down for you to show you the complexity of this. We can take a cell, a living cell, and we can break it into its component parts. We'll leave the DNA intact so you have the code. We'll leave the polysaccharides, the sugar so you have the code. We'll leave all the enzymes intact so all of those have their inherent code. So we won't decompose it to the basic amino acid units. We'll leave it. But we can pull that apart, take apart the lipid bilayer, put those lipids in a bottle. And now you say to the chemist, to the biochemist, here's all the components. Can you just put this back together again for me, please?
Answer.
Nobody would even say that they know how to go about trying to get this thing to work. The components here, and I'm not even talking, you can say with a bacterium, the simplest bacterium, just put this back together for me. So let's even make it more simple for them. Let's just take the simplest cell we have, and it just dies. Now bring it back to life, because everything is, resurrection has got to be easier than ab initio synthesis. This is why for God, a resurrection is not very hard. I mean, compared,
you already created in the first place.
Yeah, from all these diverse molecules.
Right.
Everything's just about in place. We don't even know how to well define what we just lost when I say it died. What is it we lost when I say it died? And it died a nanosecond ago. Now bring it back. We don't even know how to get it started again. That's how clueless we are on the origin of life.
Now, you mentioned earlier, and I want to maybe focus on this for a minute, and that is this issue of the God of the gaps, because it's common, and I've seen this multiple times in debates with skeptics and atheists that, well, Dinesh, your reasoning with the God of the gaps, because we don't know something, you are inferring God must have done it. I think what you're saying is, I'm not inferring that. Let's just begin by acknowledging what we don't know. And then that opens the door to us starting to think about how this might have come to be. And in that, I don't see what is wrong in considering, among others perhaps, the God explanation. Well, what is particularly out of bounds in doing that? In other words, you have this. You have the cell. It's got all this information. I mean, we know from normal human interaction that we can spot intelligence and intentionality in ordinary life, right? If I see a head rolling around in an alley, I know it didn't just end up there. Somebody killed that guy, or he committed suicide. There's some action, intentional action, that produced that result. Or if I see someone plotting some kind of a burglary, I can see from the intelligence that went into that wasn't done by a stone. Some human being figured that out, because plotting requires intelligence. And here we have a cell which shows all the indications of a great deal of creative ingenuity going into producing it. So what's wrong in saying, if aliens didn't make that, God maybe did?
Yeah, so I've never brought that in, because as soon as I bring that in, they will dismiss me. So what I've said is that I presume one day we will learn how to make life in the lab. And the reason I say that is this, because if you had asked a man, say in 1600, would we ever be able to go fly to the moon and walk around on it and come back? He'd be like, what are you talking about? You didn't even have flight, let alone space flight. I mean, this would have been incomprehensible. But humans are really ingenious. And they figured out how to fly. They figured out how to do space flight. They figured out how to land on the moon and come back. And so we don't know what humans are going to come up with. These discussions that you and I are having, I mean, people are going to look back, if YouTube still keeps these up in perpetuity, in 500 years and go, those people were pretty clueless. I mean,
look what we can do now.
Everybody in kindergarten knows how to do this. And so I always say, I presume one day we will solve it. That right away separates me from God of the gaps. Because God of the gaps is saying, we can't do it, and it says, and therefore God must have done it. I'm not saying that at all. We don't know how to do it yet. So I always say, and I challenge Steve Benner on this, why do you say God of the gaps? He says, well, if you say it's not going to be solved for 500 years, that's essentially saying we can't do it, so it's God of the gaps. I said, no. I mean, you see the sleight of hand of these guys. Because if they can say God of the gaps, for me, they'll dismiss me. And the one derogatory word you're allowed to continue to call people in the university without being canceled is creationist. He's a creationist. We don't have to deal with him. Tour's a creationist. We don't have to deal with him. So I never bring creation into this argument. Let's deal with the science. Now, if you want to talk where I stand in the Bible, in 6 days, God made the heavens and the earth and all that is in them. I don't quite know the length of the days, where we were in the expansion of the universe, but God created fundamentally everything. But I'm not bringing that into this. I presume one day we'll be able to make it, and I'm saying you're nowhere close.
I mean, what's telling to me about all this is that, there is a general assumption that people who are religious are dogmatic because they accept claims from Revelation. They have a sacred scripture. I believe it because the Bible says it, and so on. And science, we hear, is a better way of learning because science is undogmatic. Science doesn't purport to know things it doesn't know. Science is always willing to revise and adjust its beliefs when new evidence comes in. And yet, from all you're saying, the scientists are just as dug in many ways as the believer. They're just as dogmatic. And in some ways, it's even worse because most believers will at least say, well, I have faith. In other words, I acknowledge that I'm believing this on the basis of revelation. The early writers of the Hebrew scriptures basically said, God told me, and we're writing it down. Whereas the scientists claim to know based upon this tried and true method of hypothesis, experiment, verification, testing, and you always get the idea that they're not going beyond the evidence. But I think what you're showing is that in many areas, and we've seen this in other areas beyond what we've been talking about, they often do.
Yeah, they go beyond the evidence all the time. I mean, I'll give you an example here. So if for example, dark matter. So it is presumed that 70, 80% of all matter is dark matter. What does that mean? You can't detect it. You can't measure it, but it's there. You're telling me you can't measure it? No one's ever measured it. No one's ever detected it. Yet all scientists hold on to this idea of 70 or 80% of all the matter that's around it. I can't see. I can't even detect. No way. They get it by difference. They look at what's happened in the universe. There's a lot of matter that's unaccounted for, so they're calling it dark matter. Dark energy. Dark energy, again, there's energy they can't detect, but by difference, they're saying this energy must exist. This is something that is totally untestable. Now, maybe one day we'll be able to detect it, but totally untestable. And they hold on to this dogmatically. And I'm not disagreeing with them. I'm just saying, yes, there's a whole lot of faith that you guys have. The other argument is, you tell me I have God of the gaps, you have time of the gaps. If given enough time, it will happen. Time is your enemy when it comes to chemistry. What you do when you have, for example, RNA, say you had a 600 mer of RNA, which is very short. How long does that last? If we say in a lab, in water, that could last 100, its half-life would be 100 days. So instead of having, we talk in moles, but people don't know what moles is. Instead of having 100 molecules, we have now 50 molecules after 100 days. So it cut in half. That's the half-life. When you talk about one molecule, if say some molecule form that could prescribe being good for something, even though the chances of that are statistically less, we don't have enough time in the universe to have that, but let's say you found it. That molecule of RNA, that 600 mer, you take the half-life, you convert it to a probability, and it lasts 3 hours. 3 hours is the twinkling of an eye in geological time frames. So if it formed, it now has to find all the substrates for it to act upon. But for it to have formed, it couldn't have had any of those other substrates around because they would have interfered with it forming. So time is your worst enemy when it comes to origin of life, because everything you make falls apart very rapidly. And so they will say, well, given enough time, enough of these. And so for them, it's time of the gaps.
I mean, this is an absolutely key point, because I think one of the reasons that a lot of people, perhaps me included, have tended to believe some fairly fantastical things is by extending the time scale. In other words, the idea that there could be even common descent, let's just say between you and me and a fruit fly, is laughable on the face of it, right? And so it's only by saying, well, this has occurred over eons and eons and eons. Of course, we didn't go from you to a fruit fly, Dinesh. We went from a fruit fly to a little different type of flying creature. And then slowly we got to the fishes. And then the fishes came on land and became reptiles and amphibians. And then we got to mammals. So all of it is very stretched out. I think what you're saying is that, at least let's focus on the origin of life here. Let's just say by some chance you got the right kind of element. Boom, it's there. But it's not going to be sticking around for all that long, and it needs all these other elements to create life. And if those other elements aren't already present, it's just not going to happen.
Yeah, and if it happens to be in Houston, one of these molecules, and the other molecules you want are in Los Angeles, how did they get together? How did they get together? And if you say, well, they had to have been here in Houston too, then you have to invoke things that the concentration would have had to been so high on Earth that it was raining these things down, and nobody will buy that. So the concentration arguments kill you. All these things in vents that they say in vents in the ocean, these happened. Proteins racemize, they decompose at these temperatures. They want the temperatures to induce the reactions, but the temperatures decompose the reactions, so that now they're hard pressed. And then there's the dilution factor. Boom, it goes into the ocean. How do these two molecules find each other? So you need to run your reactions at a sufficiently high concentration. And you do it in an enclosed flask so they can eventually find each other and bump up. If you do it too dilute, your reactions never occur because they never found each other. You do it in an ocean, they never find each other. So every argument they have, I'm telling you, everything breaks down under scrutiny of chemistry. But what's interesting, getting back to your former point, they are beginning to see this. You look at biologists, they're being confronted with what they are saying, boy, this thing looks designed. Even some other people have said, it looks designed, but they're talking about design. The physicists, where you have the fine tuning, you have these constants and you have things that are so exact, they're like, how did it get so exact? There's the fine tuning, which certainly looks like, and then you have the chemists going back and saying, look at these molecules. So I think we keep pushing this thing, and you're going to go back and you're going to see that something's going on. Something is happening here.
I mean, with the fine tuning, we're not talking about even normal improbability, right? I think I've seen someone say this is something like shooting an arrow across the entire distance of the universe and hitting a target at the bullseye level. So you're talking about fantastically improbable odds of this kind of thing somehow occurring by chance. And therefore, I think our conclusion here is this, and that is that there was an argument from design advanced in a fairly simple way in the early 19th century. Maybe that version of the argument has to be modified, or maybe even has been discredited. But I think what we're saying is that there is a new argument from design arising out of the most complex knowledge of modern physics, modern cosmology, chemistry, and biology. And this is an argument that it's very difficult for the skeptics and the atheists to refute, because it is standing foursquare in the middle of the most cutting-edge findings of modern science.
Yes, I would agree. And that's why I think that when a man like Lee Cronin comes and says that information came from outside the cell, that's huge. Because information is the source of this. And when you look in the scriptures and it says, in the beginning was the Word, information.
Logos.
And the Word was with God, and the Word was God. I mean, this is amazing. He starts with information. In the beginning was the Word. This information, Word is information. It's a bunch of letters strung together that give you now information. God is information. The Word was with God, and the Word was God. And the Word became flesh and dwelt among us. and we saw his glory. Glory is of the only begotten from the Father, full of grace and truth. This is amazing. It starts with the information, and that's what they're saying. Somewhere that information came.
And I think if I can close on this note, I think it teaches us as Christians not to be resistant to science or afraid of it. I think you get a little bit of both in some elements of Christianity. Somehow the belief that these scientists are up to some kind of atheist hocus pocus, but rather to approach science and to approach the scriptures with that sense of wonder, but also a kind of critical mind to see the ways in which these two books, if I can say, that God has written, the book of Scripture, but also the book of nature, these books are telling us the same story in different ways.
I think so. Thank you for joining me today. If you could give us a like, share, or podcast review, we would appreciate it. If you have any questions, you could send them to ask@jesusandscience.org, and we'll try to answer some of those questions in an upcoming video. And if you do not believe in the physical resurrection of Jesus Christ, and you want to hear about why I believe, send me an email to tour@drjamestour.org, and we'll get together by Zoom, and I'll share with you why I embrace the resurrection of Jesus Christ.